Dear learners, welcome to this online lecture series of Swayam course, that is Food Fundamentals and Chemistry. In previous videos, you have learned about the different food groups, basics of food, water and carbohydrate. Today we will be discussing about the protein. Just imagine your body as a big building made up of brick. For to build any big building, a strong building, we require bricks. In a just similar way, the protein is act as bricks to body build and repair. So, guys, if we are gym enthusiastic or any gym enthusiastic, they are always worry about their body building and body fitness. So, protein is very important nutrient to build the body, to repair the body. So, we will discuss today about the protein with the following objective, that is learning objective for this video, that is to describe the different levels of protein structure and classification, to comprehend the functionality of protein in food system as an enzyme, structural components, emulsifier, texturizing agent, etc. To understand the application of protein and protein based ingredient in food processing and preparation. This protein is the most abundant organic molecule in our body, and it is made up of carbon, hydrogen, oxygen, nitrogen and sometimes sulfur. It occurs in every part of the cell and constitute about 50% of cellular dry weight. So, this protein word comes from Greek word, that is proteus, meaning first or primary, or we can say important. So, protein consists of the amino acids. So, that means protein is made up of amino acid. The amino acid is linked by the peptide bond. There are 20 amino acids found in the structure of protein. The protein is not only the important as a constituent of food, but it also perform the different function in food processing and preparation. Nutritionally, the protein provides energy and it helps in body building, maintenance and tissue repair. Some protein speed up the chemical reaction, while others play a role in defense, storage, transport, cellular communication, movement and structural support. It gives structural support to the body. So, where we will get this protein? So, protein is the constituents of most of the molecular or nuclear material of the cell, that is DNA, RNAs we can say, and it occurs in dried seeds of plants more than in the plant tissues. So, there are three main sources of protein, which includes we can get protein from the plant, we can get protein from the animal and we can get protein from the microbial sources also. So, let us see how much amount of protein present in the different food. So, if we will see some of the pulses, oil seeds and fresh vegetables and fresh fruits we can say. So, the higher amount of proteins are present in oil seeds and dal and pulses. This Bengal gram we can see almost 20% of the protein is there, black gram, green gram, lentils, dry beans, then we can say soya beans, groundnuts, sesame, cotton seeds, sunflower seeds, that are the good sources of protein. So, they have more amount of protein is there, that is almost about more than 20% of protein is there. But at the same time, when we will see the fresh fruits and vegetables, the fresh fruits having less amount of protein, we will see fresh bean, that is only 2% of protein is there. In carrot, only 1% of protein is there. In case of fresh peas, that is 6% of protein is there. So, this is the amount of protein we will get from dal and pulses. So, in a similar way, there are animal and microbial sources of proteins are there. We will get more amount of protein from meat, milk, eggs, fish. As compared to the milk, we will get more protein in meat, eggs and fish. That is almost more than 10% protein is there. So, in case of meat, we will get 18 to 22% of the protein is there, that is good quality of protein is there. Then we will get the protein from the microbial sources, that is single cell protein is there, that protein is more than 40% protein we will get from the single cell protein. So, after this we have mentioned that protein is made up of amino acid. So, what exactly this amino acid is there? So, amino acid consists of each amino acid consists of the 4 components are there, that is one central carbon atom, then another one that is amino group, that is NH2 group is there, and then carboxyl group, that is COOH group, and another one that is R side chain, that is side chain R group is there. So, whenever we are talking about this amino acid, the amino group, that is basic one, and another group is there, that is carboxyl group, that is acidic in nature. So, difference in side chain, whatever the side chain is there, R side chain, difference in that R side chain distinguish each amino acid, that is whatever the 20 amino acid found in the food or found in the protein, that are difference between that amino acid, that is based upon the side R chain is there. The amino acid is termed as alpha amino acid if both the carbon and both the carboxyl and amino group are attached to the same carbon atom, that time this we can say that is alpha amino acid. And amino acid mostly exist in ionized form in the biological system. If we will see that general structure and whatever the existence of that, whatever the amino acid is there, that is in ion form. We have already discussed about this amino acid is linked together with the help of peptide bond, and then after that there is formation of polypeptide chain is there, and we will get that polypeptide chain of amino acid means protein. So, how this we can say polypeptide bond will form, we will see the linking of 2 amino acids with each other by the elimination of H2O molecule resulting in the formation of peptide bond. If we will see this one in an image also, we will get that from one amino acid, that is OH group, and from another amino acid, that is hydrogen bond, that is 2 H atom and 1 oxygen atom will removed from the structure of this 2 amino acids, and we will get the formation of peptide bond, and we will get the polypeptide chain of amino acids. When these amino acids link together, you will have the N terminal, that is side with the amino group on the left hand, and C terminal on right hand side. So, at the molecular level, the peptide bond is formed through a dehydration reaction, as we have said that water molecule, that is H2O is eliminated from the structure, that is means the dehydration reaction is there. As shown in the image, 2 amino acids are able to bond together when 2 hydrogen and oxygen are removed from the molecule, we will get this formation of peptide bond is there. Then we will move to another portion of this protein, classification of protein is there. Protein have the wide structural and functional diversity. So, it is difficult to classify on the basis of a single property characteristics or however, the following are some common classification are there, classified on the basis of hydrolysis of product of hydrolysis, then shape and structure, then biological function. So, on this majorly on these 3 bases protein are classified. Let us understand the classification of protein on the basis of these 3 criteria. So, classification on the basis of product of hydrolysis into the 3 groups, that is simple protein, then conjugated protein and derived protein. Those protein are whatever the simple protein are there, those proteins are only made up of amino acids. It is 2 types of simple protein, that is one globular protein that is in a spherical shape, or another one is a fibrous protein, that is thread like shape is there. We can say in case of this fibrous protein, whatever the protein available in the hairs, that will be included under the fibrous protein category. So, another group is there, that is conjugated protein. They are made up of amino acid and prosthetic group, that is we can say non-amino acid group. Example, phosphoprotein is there, nucleoprotein is there, glycoprotein is there, mucoprotein is there, chromoprotein is there, lipoprotein is there, metalloprotein is there. For example, if the protein is attached with the, we can say, metals or metal ions, in that case that is metalloprotein is there. In case of phosphorus group is attached with the protein, that we can say phosphoprotein is there. And third one category of this protein based upon the product of hydrolysis, that is derived protein. They are made up of by water removal, or we can say small peptide form like peptones break into the amino acids. So, this kind of classification are there on the basis of product of hydrolysis. Then we will move to another classification, that is based upon shape of protein, and on the base of shape of protein, the protein is classified into 2 groups, that is fibrous protein, and another group is there, that is globular protein. Fibrous protein have no tertiary structure involved into it. They are made up of primary and secondary structure. They are insoluble in water. They are linear. We can say they are sheet-like in shape, and they have structural role. Example, keratin in hair and nail, actin and myosin protein in the muscle cells, collagen in the skin. So, this kind of protein are the part of fibrous protein. Then another group based upon the shape, that is globular protein. They have tertiary structure, globular, that is spherical arrangement is there. That protein is soluble in water. They are associated with the metabolic processes. Example, hemoglobin, myoglobin, egg white, enzymes, antibiotics. These are the some examples of globular protein. So, then we will come to the next classification, that is we can say protein plays various role, various function in the body of the living organism, and based upon that function protein are classified as enzymatic protein. Whatever the proteins are there, they act as enzyme and they accelerate the chemical reaction. So, whatever the protein is there, that protein called as enzymatic protein. Like we can say example, digestive enzymes catalyze the hydrolysis of the bond in the food molecule. So, whatever the proteins are there that perform the chemical reaction, that considered as enzymatic proteins. Then storage protein, storage of amino acids are there. Example, casein in the milk is the major source of amino acid for the baby mammals. Plant have storage protein in their seeds. Albumin is the protein in egg white used as an amino acid source for developing embryo. These are some common example of storage protein. Then hormonal proteins are there. Coordination of organisms activity are the function of that hormonal enzymes. Example, insulin. Insulin is a hormone secreted by the pancreas, causes other tissue to take up glucose and thus regulating body sugar concentration. This is a hormonal protein, they perform this function. Contractile and motor protein are their function, that is movement of the tissues or movement of the person. Example, motor protein are responsible for the individuals of this flagella and cilia. Actin and myosin are the protein responsible for the contraction of the muscle. So, apart from this, defense protein are there, they are protective against the different diseases, like for example, antibodies are inactivate and help to destroy the viruses and bacteria in the body. Then another type of the protein is functional protein is there, that is transport protein, transport of substances into the body. For example, hemoglobin, the iron containing protein of vertebrate blood transport oxygen from the lungs to other part of the body. So, other protein transport molecule across the membrane. So, those are proteins called as a transport protein. Then after that, receptor protein, response of cell to the chemical stimuli. For example, receptor built into the membrane of nerve cell detects signaling molecule released by the other nerve cell. Then after that, structural protein is there, that whatever the structural protein that support is the main function of that protein, and the example of the structural protein is keratin in the keratin is a protein in the hairs, horns, feathers and other skin appendages. Insect and spiders use silk fiber to make their webs respectively, and collagen and elastin protein provide a fibrous framework in the animal's connective tissues. So, these are the functional protein that perform the different function in our bodies. Then structure of protein is the main basis for any functional property or any characteristic shown by the protein, that is all depend upon the structure of protein. So, there are 4 level of structure of proteins are there. Proteins are the polymer of L alpha amino acids, we know that one. The structure of protein is rather complex, which can be divided into the 4 level of organization, as we have already said that one 4 level of protein structure, that is primary structure is there and secondary structure is there, tertiary structure is there and quaternary structure is there. So, we will let us see the one by one. So, this primary structure is nothing but a normal chain of polypeptides, and we can say that is whatever the amino acid is there in a primary structure, that is arranged in a linear sequence. It depends on number and sequence of amino acids. The amino acids are connected or stabilized by the linkage that is known as, we can say, covalent bond, mainly includes peptide and occasionally disulfide bonds are there, and example of this primary structure, that is primary structure is seen in the insulin. So, this is one of the example in which the primary structure is there. So, then after that we will move to the secondary structure. If you will see this image, particularly secondary structure of protein, that is twisting or folding of linear polypeptide chain, that is primary structure, is referred to as secondary structure. 2 types of secondary structures are there, that is one alpha helix and beta helix are mainly identified into the protein, secondary protein structure. If we will see the alpha helix structure was proposed by Pauling and Corey in 1951. It is a most common spiral structure of protein around the central axis, that is alpha helix is present. So, it has a rigid arrangement of polypeptide chain. So, the structure is stabilized by the hydrogen bond formed between hydrogen atom attached to the peptide N and O atom attached to the peptide C. So, this is the structure of we can say alpha helix structure is stabilized by this hydrogen bond, and after that all the peptide bond except first and last in polypeptide chain participate in hydrogen bonding during this structure. So, each turn of alpha helix contains almost 3.6 amino acid, each of turn travel distance of 0.54 nanometer and the spacing of each amino acid is 0.15 nanometer. So, the structure is destabilized by whatever the structure is there, that is alpha helix structure, that is destabilized by proline and glycine. Presence of negative, that is acidic, and positive charges of amino acid, that is basic, which replay each other, and this also destabilize the alpha helix structure. The alpha helix structure is observed in myoglobin and hemomyoglobin. Hemomyoglobin structure and this myoglobin structure are the example of alpha helix structure. Let us see the another secondary structure, that is beta pleated structure or sheet structure is there. This is the second type of structure, that is after alpha, hence that is beta structure is there, proposed by the Pauling and Corey. Beta pleated sheets are composed of 2 or more segments of fully extended peptide chains. In the beta sheets, the hydrogen bond are formed between neighboring segment of the polypeptide chain. So, beta pleated extended in the zigzag manner. If you will see the image, we will get that one. So, whatever the this one, the polypeptide chain in a beta sheet may be parallel. Whatever the image B is there, you can check see that image, that is parallel in manner in the same direction, or anti-parallel with respect to N and C terminal. That anti-parallel we can see in the image C. So, beta pleated sheet may be formed either by the separate polypeptide chain, that is H bond are interchange, or a single polypeptide chain that is folding back on to itself. So, the H bond are intra-chain bonds are there. So, this is about the beta pleated sheet structure of the secondary structure of protein. Then we will move to tertiary and quaternary structure. The tertiary structure, it is formed by folding of secondary structure to a 3D orientation. So, it is stabilized by hydrophobic, H bond, electrostatic forces and van der Waals forces are there. They involve single polypeptide chain. So, this is about the tertiary structure, and when we will come to quaternary structure, this structure involves 2 or more than 2 polypeptide structure. Besides the hydrogen bond, disulfide bond, ionic interaction, electrostatic bond, hydrophobic bond interaction, van der Waals forces contribute to the tertiary structure of the protein, contribute to the quaternary structure of protein. Example, hemoglobin, aspartate, tricarboxylase, lactate dehydrogenase are the example of quaternary structure. Then we will see the physicochemical properties of protein. Physical properties includes solubility. Globular protein are more soluble in aqueous medium than the fibrous protein such as keratin. Solubility behavior of protein is influenced by different factors, that is nature of solvent, pH, temperature, etc. The protein forms dispersion rather than true solution due to the large molecular size. Amphoteric nature, protein has amphoteric nature, that is just because of the amino acid which contains both acidic and basic groups, they can donate a proton or accept a proton, hence amino acid are regarded as ampholytes. Then we will come to another important property, that is isoelectric pH. The isoelectric pH is the pH at which number of positive charges equal to the number of negative charges and overall charge on the amino acid is 0. At this point, when subjected to the electric field, protein do not move either towards the anode or cathode. Hence, this property is used to isolate the protein. Then precipitation is another property. Protein may be precipitated without getting denatured at the isoelectric pH in the presence of salt of heavy metals or in the presence of non-polar solvent like chloroform. In the process, the electrostatic attraction between the ions of protein get enhanced and thus facilitating their aggregation and precipitation is there. Optical activity is there. All protein solutions rotate the plane of polarized light on the left or to the right. All of the amino acid except glycine possess optical isomers due to the presence of asymmetrical carbon atoms. Then another property is there, denaturation of the protein. It is a phenomena of disrupting secondary structure, tertiary structure and quaternary structure of the protein. Denaturation involves change in physical, chemical, biological properties of protein. Denaturation may cause by physical, chemical reagents such as heat, acid, alkalis, organic solvents, salt, heavy metals and detergents. So, another we can move to the chemical properties of the protein, and chemical properties of protein is due to carboxyl group, amino acid group and R side chain. So, reactions of protein due to the COH groups, amino acid forms salt with the bases and ester with the alcohols. Then decarboxylation is there, amino acid undergo decarboxylation to produce the corresponding amines, and another property is their reaction with ammonia. The carboxyl group of dicarboxylic amino acids react with the NH3 to form amides. So, then after that reactions due to the NH2 group, the amino acid behave as a basis and combine with the acid. Example we can say HCl and it forms the salt. The reaction with ninhydrine, the alpha amino acid react with the ninhydrine to form purple, blue and pink color complex, that is we can say ruminants purple complex is there. Then transamination is there, another property, chemical property, that is transfer of amino group from amino acid to keto acid to form a new amino acid. That is very important reaction in the amino acid metabolism. Then another one, oxidative deamination is there, that is amino acid undergo oxidative deamination to liberate free ammonia. This is one of the important chemical reaction is there. Then color reactions are there. We are discussing here only two representative color reactions, that is one biuret test is there. We will use this biuret test for the qualitative and quantitative determination of the protein concentration. So, when the peptides or protein treated with alkaline copper sulfate solution which produce a characteristic purple color, the color deepens, whatever the color is there, that deepens as the number of peptide bond increase into the protein, and protein produces a deep blue violet color due to the probable formation of coordination complex is there. All protein and peptide except dipeptides respond to the this reaction. Then xanthoprotic test is there. On boiling with the concentrated HNO3, yellow color develops due to the presence of benzene ring. So, this reaction is due to the nitration of phenyl ring, presence of tryptophan, PHE and TRP, this whatever the specific amino acid is there, to yield yellow substitution product which turns orange upon addition of alkali. So, this color xanthoprotic test is there. These are the some chemical properties of we can say protein is there. Then we will move to the functional properties of protein. You are aware that functional properties of food constituents refers to their attributes that make them useful for the food besides their nutritionally important. These are the physico-chemical properties which influence the characteristics of food like texture, taste, appearance, etc. So, what are that properties? That properties are emulsification, viscosity, solubility, we can say formation of gel, binding to the fat and water molecule. Understanding these properties help food technologists to improve the apparent taste and texture of the food by exploring them. Like solubility, that solubility promote the absorption and digestion of food, as we have already seen the solubility. Emulsification capacity or emulsifying ability that aids in digestion and absorption of some types of nutrient, example fats. So, gelling capacity is there is gel formation is there, gelation we can say, gelation of protein is there, gelling property that enhance the texture of the food. Then after water holding capacity is there, that water holding capacity promote the absorption and digestion of food is there. Then foaming ability, foaming ability is responsible for proper food texture and appeal also, that enhances the palatability of the food also. Adhesive property is there that increases stickiness of the food. Then after that another property is there, that is functional property, viscosity is there, and due to the viscosity whatever the protein is there, that protein acts as a thickening agent into the certain types of the food material. Then we will come to the our very important portion, that is application of protein ingredients or protein. So, the different protein ingredients are prepared, or we can say extracted, like soya protein concentrate is there, one of the important ingredient is there. It contains not less than 70% protein on the moisture free basis. The most important application of the soya protein concentrate in food industry, that is primarily in a commuted meat, poultry and fish products like patties, sausages and fish sticks, etc. The soya protein isolate increases the water and fat retention in this product. Beside this, the soya protein concentrate are also used as the stabilized dispersion in the milk, beverages and simulated dairy products like sour cream analogs. Then another protein ingredient, that is soya protein isolate is there. Soya protein isolate contains at least 90% of protein on the moisture free basis. The major food application of this soya protein isolate are in the meat products like the meat replacer, in seafoods like fish sausages, and in cereal, bakery and dairy products. Whey protein concentrate, that is WPC, The whey protein concentrate finds extensive application in baked foods and contributes toward the product browning, crust and flavor development. Concentrate with high viscosity are used in ice cream, processed cheese and fresh dairy applications. Then after that, protein hydrolysates. The protein hydrolysates are used in soup, sausages and are more important for the peoples who cannot digest solid foods. So those are some important application of protein ingredients. Then we will move to conclusion part. So this lecture covered the basic concept of protein, classification, structure and properties. Major physical properties such as color, taste, shape, size of protein, their colloidal nature, denaturation, solubility, optical activity are also discussed in this video lecture. Major chemical properties such as reactions involving COOH, NH2 group and selected color reactions of protein are also discussed in this video lecture. Toward the end of this video lecture, the application of protein based ingredient such as protein isolate, concentrate and hydrolysis are also addressed. We hope that you have had a rewarding learning experience. We wish you a grand success in your further academic endeavors. Thank you.